Hi everyone, today we are going to learn about basics of stem cells. These are the snapshots of UPSC prelims and main syllabus. The highlighted portion shows the importance of this topic. If you are from biological background, you can expect questions under stem cells in interview. Okay, let's start our session. The stem cells are the cells which has the ability to differentiate into any type of cell. For example, it can turn into a nerve cell, brain cell, kidney cell. Before going to classification, we need to know about the term called potency. Potency is nothing but its ability to differentiate into different cells. Stem cells are classified into four types based on their differentiating ability. First one is totipotency, second one is pluripotency, third one is multipotency, and fourth one is unipotency. Totipotent stem cells are the cells which has the ability to develop into the whole organism. Example, plant cells. All the cells in the plant are totipotent. By taking a tissue from the leaf through tissue culture, you can create the entire plant. This is called as totipotency. Pluripotent cells can develop into different organs, example brain cells, nerve cells, lung cells, etc. But they cannot develop into the whole body. Third one is multipotent stem cells. Multipotent stem cells can differentiate into only certain type of tissues, that is closed related cells. Example, hematopoietic stem cells. Hematopoietic stem cells can develop into a RBC, WBC or platelets. Last one is unipotent cells. Unipotent cells are the cells that can develop into a only one type of cell. Second classification is based on origin of the stem cell. First one is embryonic stem cell, second one is adult stem cell, third one is umbilical cord stem cell, fourth one is amniotic fluid stem cell. First one is embryonic stem cells. After fertilization, zygote will turn into an embryo, and this embryo will differentiate into inner cell mass, which is called as blastocyst. These inner cell mass cells are the embryonic stem cells. These embryonic stem cells are developed into a ectoderm, mesoderm, endoderm, and a germline cells, that is sperms and egg. The ectoderm is the brain and skin, and mesoderm is the muscle cells, brain cells, bone cells, cartilages, and endoderm is the lung, gut, and the liver. These embryonic stem cells are pluripotent cells, that is, they are differentiating into a different organs but not the whole organism. Second type is adult stem cells, which is found in the organs of the organisms. These are multipotent or unipotent. For example, hepatoblast, which are the lungs stem cells. These cells can differentiate only into hepatocytes, that is the grown lung cell. This is an example of unipotency. Third one is umbilical cord blood stem cells. You can see the umbilical cord in the picture. Umbilical cord is the one which is connecting placenta with the baby. This placenta contains hematopoietic stem cells. After removing umbilical cord from the baby and before cutting placenta from the mother, the blood must be connected through the syringe, and after centrifugation it must be stored at the minus 190 degrees Celsius in liquid nitrogen. The umbilical cord blood cells or the hematopoietic stem cells. This can differentiate into RBC, WBC and platelets. Storage of umbilical cord is increasing nowadays. These umbilical cords are used to treat many diseases, including cancer, immune disorders, blood disorders. Fourth one is amniotic fluid stem cells. This fluid present in the womb where the baby floats. This protects the fetus in uterus. These cells are multipotent and pluripotent, but the disadvantage is collection process is very hard. Last one is induced pluripotent stem cells, that is triggering the four basic genes and inducing it to a stem cell. For example, we are taking a cells from lungs, which are lung cells. These are adult stem cells. By reprogramming, we are turning it into a blastocyst, which is pluripotent cells, from which it can differentiate into mesoderm, endoderm, and ectoderm. That is, it can produce different organs. The main advantage of this process is it is used for clinical trials and development biology and cell-based therapies. Thank you for listening. If you like this video, give thumbs up and subscribe to my channel.